So I thank PacBio for inviting me to present our work on rare disease and trying to find more diagnoses for kids and families suffering from rare disease at Children's Mercy Kansas City. And I'll be carrying through a discussion of several topics. First, I'll discuss the genomic risk in disease in general and what areas of disease we are investigating here in Children's Mercy. I'll discuss rare diseases in a little bit more detail, especially with respect to the challenges we currently have in diagnosing rare disease that we often think is genetic in nature. And I'll discuss next generation clinical sequencing briefly, and then through that I will present a approach that we believe will change the outlook for rare disease families in the greater Kansas City area and beyond, called Genomic Answers for Kids, and discuss some of the technological advances that we're implementing in undiagnosed disease. So properties of genomic risk differ between different types of diseases. What we are most interested in are diseases of early childhood, often present at birth. So here in the bottom of the slide, these are often rare, and genes play a large role. So most often these are diseases that are deterministic in your genes. They are inherited in a Mendelian manner in a family, or they occur through de novo mutation that is not necessarily present in parents, but it's in the germline of a parent, or occur spontaneously in the embryo. And these lead into early onset, rare, and often severe diseases. The other disease areas that we study here at Children's Mercy relate to youth onset disease, such as type 1 diabetes. I will reserve discussion on those today. And what we don't study too much in Children's Hospital and in children in general are the common diseases that are the topic of many genome-wide association studies and genetic risk scores. The rare disease problem in kids is widespread, and we do have different definitions of rare disease across the globe, but a rule of thumb would be that approximately less than 1 in 1,000 individuals would be called rare in most countries. And in the US, it's defined as diseases affecting less than 200,000 people. So individually, these diseases are rare, but collectively, they have a large impact. So up to 30 million Americans suffer from rare disease. There are many challenges for rare disease, one of which is long diagnostic odyssey. So in most kids, rare disease, getting the molecular answer, if you're ever going to get a molecular answer, takes 3 to 6 years. And as I said in the previous slide, most often these rare diseases are genetic in nature or thought to be genetic in nature. There are multiple misdiagnoses during this so-called diagnostic odyssey. And as I said, the diseases are caused by mutations in DNA, and up to 1 in 6 hospital admissions in kids in a tertiary care hospital like ours at Children's Mercy are due to rare disease. So their impact is larger than their prevalence in terms of morbidity. What we most often see here are ultra rare diseases. So these are, or what we are involved in clinical sequencing, are ultra rare diseases which occur in less than 1 in 50,000 individuals. And often there's a lot of challenges related to ultra rare diseases. We may understand something of the natural history of the disease, but usually we do not have treatments or a good idea of the mechanism of disease. But the key thing is to get the answer first in order to start to delve into the mechanistics and ultimately develop therapies for patients. For families, however, regardless of therapies, ending the diagnostic odyssey is a key point, and providing this DNA diagnosis is often increasing the family well-being. It allows the family to reach out to other families with similar rare disease. It allows the providers to tell the family what to expect and what sort of special needs their child may have. So it's always impactful, and we know this from our work with families directly in our research program, that despite the lack of, or the relative lack of treatments, the diagnosis and reaching it as soon as possible is always important. At Children's Mercy, Kansas City, we do clinical whole genome sequencing, and the clinical lab has been doing within our hospital this for several years. We require insurance pre-authorization. That is a barrier for some kids and families with rare disease. Genetic counseling is a critical need for families, and it's often limiting in terms of volumes of testing. What we do in practice is we sequence a child and both parents, and I'll show you later why that is important. And we report out according to ACMG criteria: pathogenic, likely pathogenic, and variants of unknown significance. And this is done in a symptom-driven manner, so relating to the reason of referral. Historically, the diagnostic rate has been about a quarter of a patient received diagnosis. This is still somewhat outdated. Currently, we are in approximately 1/3 of patients receiving a diagnosis. And we also report, in addition to the pathogenic or likely pathogenic variants that are considered diagnostic, the clinical lab reports out variants of unknown significance. Sometimes there's other incidental findings that are actionable according to ACMG. We see that in approximately 3% of families. The key point is that the majority of kids remain undiagnosed after clinical whole genome sequencing. So why is this? Well, there are several potential reasons, some of which are shown here. So a key reason that we believe accounts for most of the undiagnosed sequencing is that the gene has simply not been yet discovered, or the phenotype associated to the gene has not been deciphered. The other reasons are that the genetic change is undetectable to the technology that is currently used in whole genome sequencing, and I'll discuss some of these findings through the work we've done with PacBio sequencing in undiagnosed disease. And obviously, we have kids with rare syndromic disease that is, despite suspicion, actually not genetic, but somehow an environmental insult that doesn't show up in germline DNA. So in our laboratory, the predictors for success in clinical next generation sequencing are described to some extent here. What we do see is that certain structured phenotypic terms, as shown here as HBO numbers, so these are from human phenotype ontology, are predictive of success in diagnosis, and these include things such as muscular weakness or global developmental delay, which are relatively commonly observed, especially in infants and toddlers, and can be useful indicators for potentially genetic underlying cause. Of course, we do see a lot of congenital anomalies too. These are usually individually rare, but when they are present, they are positive indicators of underlying gene finding by current sequencing. An important consideration for sequencing is the ability to measure both parents and the child, and it's not obviously always possible, but when it is possible, it makes a huge positive impact in ability to actually call a variant diagnostic, and therefore we always do full trio sequencing. I would note that most of the variants that are diagnosed currently in the lab are actually variants that are not seen in the parents, that have arisen in the germline genome of the patient or the kid, and that is a key diagnostic indicator. But like I said, we still have a lot to discover. So this is from my collaborator, Kim Boycott at Children's Hospital of Eastern Ontario. She has worked more than a decade in developing the rare disease program in Canada, and described many new genes. And in discussion with her, she predicts that we still have up to 10 years of gene discovery to do, since currently we are sitting up at approximately 4,000 known genes for about 6,000 diseases, and the predictions are that there are up to 6,000 genes and up to 10,000 rare diseases. So we still have many years of gene discovery at current rate. So at Children's Mercy, we want to accelerate this process in many different ways, and we have devised a program called Genomic Answers for Kids, and we want to have a comprehensive and rapid access to genomic diagnostics and sequencing, but also improve the quality of the genomic analysis in unsolved disease. So we want to move from today's situation, where we estimate that a minority of patients that actually suffer from rare disease are getting comprehensive genetic testing, and the overall diagnostic rate is less than 40%. In Genomic Answers for Kids, which we initiated in July of 2019, we aim to reach a large fraction of rare disease patients in our catchment area of greater than 5 million people. We estimate that there's about 50,000 rare disease families in our region, half of which impact kids. So we have a lofty goal of sequencing about 30,000 families by year 2026. Today we have approximately 3,000 families included in our Genomic Answers for Kids program after a little less than 2 years of operation. And in addition to increasing access, what we want to do is carry out what we call integrated genomic assessment to improve the diagnostic yields of individual tests from less than 35% to over 50%. And over time, we do want to reduce the time to diagnosis by different means, by accelerating access of primary care physicians to genomic medicine, but also the diagnostic process itself. So currently, what we employ in Genomic Answers for Kids is primarily whole genome sequencing, and we've explored multiple different options for whole genome sequencing, including long read sequencing, link read sequencing, 2 different types of PCR-free sequencing produced by Illumina and MGI, as well as exome sequencing, which is often the starting point for our analysis, since exomes are most commonly done clinically. We have developed pipelines to assess the rare variants and annotate those, and then we're using open source prediction tools that have as input, in addition to the VCF or the variant call files, they also have input from the phenotypes in the form of HBO terms that lead into a list of prioritized variants using literature. as an input in the machine learning algorithms of these approaches. And we've been pretty happy with the call rates of automated analysis. So in close to 1,100 families, we found that if expert clinical interpreter of genome called the genome negative, there was agreement in 32%. If the clinical interpreter called a candidate variant, the automated system also identified this as a top candidate variant in large fraction of cases, and then there's some areas of disagreement. But overall, we estimate that about 80% of currently diagnosable variants could be extracted relatively easily using these emerging algorithms. So June is a 4-year-old girl that was, or she was 4 when she was enrolled into our study, and she was one of the early families that was recruited into Genomic Answers for Kids. And her story is somewhat common, albeit there's one uncommon part in her story, is that she had earlier whole genome sequencing done in the neonatal period as she had significant respiratory distress at birth and multiple congenital anomalies. And she was enrolled into a study program at the time, but the gene was not found, or the mutation was not found at the time. She was then among, as I said, the first ones enrolled into our study. Her phenotype had evolved somewhat from the early phase of her life, but this is really a key issue to think about when we develop research protocols. You should never really give up on trying to re-diagnose a patient, as the phenotype is dynamic, but also the literature is dynamic. And in June's case, the literature turned out to be a real key issue. We did redo sequencing to ensure quality by PCR-free methods, and found in this PCR-free genome sequencing a deletion variant in a gene called MYRF. It was not diagnosable in 2015 as the gene was not yet discovered, and it was discovered in 2017. So this would be a typical case of diagnosed kid in our program for whom we ended diagnostic odyssey. Majority come from evolved evidence in the literature of the disease gene assertion. A smaller proportion comes from better coverage of sequencing and new sequencing methodologies. But we still want to pursue the comprehensive assessment of the genome, starting from these negative clinical genome analysis, reanalyzing them periodically to make sure that nothing new evolved in the literature. But in addition to following up the literature and reanalyzing the existing genomes, we do want to decorate the genome with as much functional data as we can from the patient's cells and tissues. And also we want to explore variants that are not seen by current short-read sequencing technologies using first-generation genome sequencing by PacBio HiFi. We also include in vitro screening by massively parallel reporter assays and related technologies to prove function of innocent-looking genetic variation. Ultimately, we believe that this cyclical process that is comprehensive will have a unique catalog of genomic variation that can be harnessed not only for individual cases, but also more widely to understand how to improve genomic diagnostics for rare disease. So the first challenge that we have is sometimes the DNA sequencing is incapable observing all the genetic variation, and we work with PacBio quite extensively in the recent year to validate and benchmark both single nucleotide variant detection and structural variant detection in our clinical samples enrolled to the Genomic Cancer for Kids study. And in this complex slide, I'm showing some key numbers. Here's a number of approximately 500,000 PacBio exclusive single nucleotide variants that are not seen by short read sequencing technologies by Illumina. Reciprocally, there is Illumina discovered single nucleotide variants not seen in PacBio sequencing. There's a slightly better specificity of the PacBio single nucleotide variants. So clearly there's a large fraction of even single nucleotide variants missed. Obviously, most of these are in repetitive regions of the genome that don't harness necessarily diagnosable variation. In structural variants, the situation is even more drastic, where we do see about 22,000 structural variants in long read genome sequencing by PacBio as compared to around 10,000. So around double the number of structural variants are detectable by PacBio in our using the current pipelines we're using and in our clinical samples. Here are some examples of key findings that were observable by PacBio sequencing. We had an inversion of collagen 5A1 exon 1 that is cause of Ehlers-Danlos syndrome, and it's the most common gene for this disease that was missed by short read genome sequencing. Similarly, we can detect compound heterozygote variants directly on genome sequencing. As I told you before, we do try to collect both parents, and that will allow us to tell for recessive variants such as this in NPC1 gene, whether or not they are compound heterozygotes or carriers of variants. But in some cases, as for this patient, the parents were unavailable, so we had to molecularly establish that the patient was actually having a homozygous or complete null of the gene. Another example is shown for this large family, in which the family had gone through several years of diagnostic odyssey, starting from this generation where the pediatric neurologist, Keith Kaufman, in our hospital observed a complex phenotype across siblings in 2 branches of the family, where we had both ataxia, seizures, and dystonia, and also the sister and brother, the parents of these cousins, were also affected mildly with lesser degree of severity of the neurological symptoms. So we enrolled the whole family into the study and applied PacBio long read sequencing, and we found an insertion variant in the intron of STAR-D7 gene that had been very recently described in the literature. So this actually the same month as we found this by long read sequencing. So this is a repeat expansion of about 1,000 base pairs that is completely invisible to short read sequencing. This gene was also reported in an Italian family to have repeat expansion in the same part of the intron and causing related neurological disorder. So this is an example where short read sequences fall short from being able to do the diagnostics, and we are developing a separate directed analysis for repeat expansions, which are also somewhat a challenge for out of the box long read sequence interpretation due to the variability in the repeat content. The last part that is revealed by long-read PacBio sequencing is the non-referenced genome. So if you think of a reference genome here on top, we do have like the build 38 genome that we're currently using in our clinical and research sequencing as our reference genome. We know that there are pieces of this pieces of the genome that are missed by current alignments to the reference genome, and by aligning de novo the genome from the long reads or building a personal genome, one can find these yellow bits that are in addition to the reference genome. So we are systematically annotating genetic variation, the transcript content, the regulatory regions, as well as the overall characteristics of these personalized bits in the genome across all our patient samples. And here are examples of 3 patients in which I've shown in the x-axis the length of the personalized bits and the number of the personalized bits that are seen in addition to the build 38 genome. And the key part here are these red colored portions of these bars, which represent rare personal content of the genome. So these are potentially new disease variants or harness new disease variation that is relatively rare in population. And we've shown that in these rare personalized bits of the genome, here's one example, we do have expressed genes. So this is a personalized contig from a patient in which we also had a brain sample available. This was a patient with intractable epilepsy treated in our hospital by epilepsy surgery, and we scavenged a excised piece of brain in this patient and carried out transcriptome sequencing using PacBio isoseq methodology, and you see clearly that there are exon intron structures predicted by the isoseq methodology, and these map to a piece of DNA that is not present in the current reference. So we estimate that there's over 100 genes in the non-referenced DNA assessed by these methods. The second challenge that I go through more briefly is that we currently, according to the ACMG criteria, we generally don't look at variation that impacts gene regulation or RNA in a way that is not easily interpretable from canonical splice sites or by direct sequencing of patient tissue in cases where that's available. So we want to explore the ability to actually routinely characterize the RNA and gene regulation in parallel with the DNA sequencing to be able to actually interpret the whole genome and not only the coding genome, which represents only a small proportion of constrained human genome. So we're building out for genomic cancer for kids patients these pediatric tissue atlases. Here's an example of several tissues from different patients. So approximately 10% of our patients undergo invasive procedures, and these provide us opportunities to actually examine the diseased tissue, as I showed in the previous slide. For epilepsy, we had a diseased brain specimen, and we could examine directly the RNA content there. Here's an example of single cell sequencing in RNA from multiple tissues and an inset of brain cells sequenced in living rare disease patients from these clinical specimens. What we can see by these approaches is, for example, unannotated exons in well-defined disease genes, just such as FMR1, that is the causal disease gene for Fragile X. And we found through direct isoseq in brain samples that there are minority species of transcripts with the constrained exon that is not currently annotated in databases. So clearly there's some potential that that new species of the genes are actually mutated in undiagnosed disease. And we can look at regulation either as a regulatory element activation or, as shown here, as regulatory looping through using HiFi approaches. What we've done systematically is generate single cell sequencing from patient samples. And while we don't believe that it always leads into the defective gene, it can reveal groups of patients based on very fine genomic features. So here's a typical single cell RNA sequencing experiment with each of these dots represents a single cell transcriptome, and this would be a normal finding. But then we see some patient samples that fall off from this normal distribution of single cell transcriptomes, and obviously sometimes these may group, which may tell us about a shared molecular mechanism and lead us to the trail of a missed diagnosis. The same shown here as a heat map across different ATAC-seq or open chromatin sequencing clusters in approximately 400 rare disease patients. And you do see some patients that have specific, or groups of patients that have specific features in their chromatin that may tell us about the underlying mutational signatures. Here's an example of an individual case of a altered chromatin signal that is due to a mutation, or actually a structural variant in the APC gene. And this could be detected by observing a low signal from the promoter of the APC1 gene in a kid that didn't have diagnostic sequencing, but was then diagnosed by a microarray to have a deletion of the region, leading to half of the activity of the promoter in the APC1 gene and the disease phenotype of FAP. So obviously, we are limited by the available samples. As I said, we scavenge patient samples from surgeries that they go through for therapeutic or diagnostic purposes, and this is successful in a small minority of patients. What we really want to be able to do is turn the blood cells of each patient that are always available to us into different cell lineages, and Scott Younger's lab at the Genome Center at CMKC is developing this approach from the genomic cancer for kids patients. Ultimately, we want to scale this up to include thousands of kids iPSCs, or induced pluripotent stem cells, in genomic profiling of single cell profiles, single cell chromatin profiles, as well as a single cell isoseq profiling, to be able to use comprehensive maps of function in parallel with the unsolved genetic variation that remain in these patients. The final challenge that we have is the undiscovered genes and how to discover them more efficiently. So the key here is to share data across the rare disease community more efficiently than we have been able to do in the past. So this includes not only sharing individual variants, but including larger and larger fractions of the genome, maybe even the whole genome sequence of many patients, along with their medical record data, and do this as globally as we can to overcome the limitation of discovery at the single center and through traditional gene matching. So we are establishing an approach to do comprehensive sequence and phenotype-based matching together with the Canadian Care for Rare program led by Kim Boycott and Mike Prudno. And the goal is really to be able to harness from 2 large-scale programs for unsolved gene sequencing as well as solved gene sequencing to discover new disease phenotypes and variants that are not discoverable at the single center. So at the end, I want to thank all the folks that have contributed to this work. So the Genomic Medicine Center at Children's Mercy, Kansas City, the large number of people involved in the work are shown here. We wouldn't be able to do this work without our clinical partners. Key clinical partner is the Division of Clinical Genetics that sees thousands of patients and is the largest referring unit in our hospital, but we do see many other divisions, clinical divisions, contributing, particularly pediatric neurology, to our family recruitment. Our international partners in the Canadian Rare Disease Program, and then PacBio team for really getting us going with the long-read sequencing and expansion of the third generation sequencing capacity, which we are now implementing as the first line follow-up for negative exome sequencing. And to end, I'll show you the picture of our newly built Children's Mercy Research Institute, shown in night lights here. And for some of you, this may resemble something that you've done perhaps in the 80s or in the 90s when you were sequencing the genomes. These are actually Sanger-like sequences from patients that have been diagnosed at Children's Mercy, Kansas City. So I'll end there and be happy to discuss. Thank you.